Here cell division and continuation life, and here the history of this cell division and continuation. First, how these scientists have worked on this topic. Let us see now. Virchow in 1821 to 1902, he proposed that cell theory. Actually, he started that in 1852. Robert Remarque, the German scientist. In 1852, German scientist, that is Robert Remarque, he observed the cell division first. Actually, this is the first attempt to understand the mechanism of cell division. And after that, in 1897, that is Fleming, in 1879, Fleming examined many kinds of animal and plant cells and selected those that showed division. So he first observed that actually that led to the discovery for chromosomes appear in double nature. Okay, here he observed the mitosis. Actually, this is the thread-like structures. Those are present in the nucleus, and the division, everything he observed, and he named them as fine threads, because mitos means fine threads. So he gave the name mitosis. And then after that, there was a sequence of events in the process of division. Even that also he observed first time, and after 10 years, these structures were named as chromosomes. Chromosomes means because these are called as colored bodies, actually the meaning, but they used many types of stains. So they gave the name as chromosomes. Actually, those are not colored bodies. Only thing is because of the uses of the number of stains. Then after that, this was actually the important discovery of chromosomes. And then after that, Rookes, Wilhelm Rookes, he proposed the chromosomes carried a different set of heritable elements. So for the first time, he observed that heritable characters are transformed from the parent to offspring. And also he proposed that longitudinal splitting. And then and one more thing, important thing he observed is equal division of these elements in the chromosomes. That also he observed. And after that, 1866, Gregor Mendel, he developed that theory, the heritable elements, how they are transformed from parent to offspring. In case of peas, he observed first time, pea plant, and he highlighted the central role of chromosomes. Actually, the central role of chromosomes was highlighted by Gregor Mendel. And in cell division, actually, the cell will be divided into two equal halves and equal number of chromosomes. This is important, and they are similar to parent, and they are diploid in nature because 2n will be continued. That's why it is a mitotic division. Duplication, we can say, duplication of chromosomes. And one more important thing, they observed for the first time that the number of chromosomes is same. They are maintained in the same way. Then next one is August Wiseman's theory, that is the hypothesis, that let us see on the board now. Yeah, now I discuss August Wiseman's hypothesis. That is from the period 1834 to 1914. Here in successive generations, actually he proposed through two he proposed his theory like this with 2 points. The very first point is, in successive generations, the individuals of the same species have the same number of chromosomes. In successive generations, the individuals of the same species have the same number of chromosomes. So here the number of chromosomes will differ from species to species. So we can say that in human beings, the number of chromosomes are different, males different, like that. So different species will have different number of chromosomes. That he observed for the first time, that is Wiseman. And the next one is successive cell divisions, the number of chromosomes always remain constant. Even this is also very important point for the further research. Okay, so what is this? In successive cell divisions, the number of chromosomes always remain constant. So the number of chromosomes are not changed. So that is mitotic division, duplication. Okay, so these 2 points, very important points for the research of this cell division. That helped very much for the future scientists. So first one is different species will have different number of chromosomes. Second one is the number of chromosomes number is same in all the offsprings. That is not, of course, offspring, because it is duplication. Okay, mitotic cell division. This helped very much for the further research. Then 2 kinds of cell divisions in the life of every individual. So every individual, all the living beings will show 2 kinds of cell divisions. One is the mitotic and the other one is meiotic. Mitotic cell division we can see in the body cells, that is somatic cells, and meiotic division in the germ cells are the sex cells. Okay, here in mitotic cell division 2n, that is diploid number of chromosomes we can see, and in meiotic division, that is reduction division, we can see the haploid number of chromosomes, that is single n, and here 2n. The next one is the chemical nature of the genetic material. Actually, what is the genetic material? That is DNA and RNA. First time they observed that something is there in the nucleus of the cell. Okay, so the material which is present in the nucleus of the cell is nothing but the DNA and RNA. This is having the chemical nature. This is nothing but the deoxyribonucleic acid. Okay, this is ribonucleic acid and deoxyribonucleic acid. So these two are present in the chromosomes, that is within the nucleus. Okay, the next one is in 1953 by James Watson and Francis Crick. Actually, Watson and Crick model we say. Okay, so this is here both of them observed that mitosis, this that is the duplication, takes place in all the body cells. Okay, it retains the same number of chromosomes, because in mitosis the number of chromosomes should not be changed, because the same, suppose if any body part we get hurt, then immediately mitosis will take place. If any injury is there, then immediately bleeding will be there. After some time the bleeding will be stopped. Okay, so when the this is stopped, that means mitosis already has taken place. So the number of cells will be repaired. Actually, the body repair for this, and for the growth of new cells, mitosis will help. So this is compulsory. So this will take place in all the body cells, that is the somatic cells of the. So there the number of chromosomes should not be changed. So the same number of chromosomes will be retained. That is duplication. That is nothing but the mitosis that they for the first time they observed and introduced that. That is discovered that Watson and Crick model. The next one is meiosis. This is reduction. Actually, this will take place in the germ cells or the sex cells. And here this chromosomal number will be halved. If the chromosomal number will not be halved, and that will be continued retained just like mitosis, if that is the case, then what happens? The number of chromosomes will be doubled. Like that, the number of chromosomes will be increased, and the symmetrical organization will not be done in the body. So here in the germ cells, compulsory meiosis should take place, because this is the sexual reproduction, and the male and female after fusion, the number of chromosomes will be same. So like that, in order to continue the race, so meiosis is very important. That is nothing but the reduction division. Now let us learn about this mitosis first in detail. Now cell division in human beings. The process of cell division is same from unicellular to multicellular animals. This you remember. So you should not think that unicellular organisms will show one type of cell division. No, not like that. All the organisms, whether it is unicellular or multicellular, the cell division, the process of cell division is same. So if you learn one thing, that is the same in all the living beings. The next one is cells in heart and brain of an individual never divide. Remember this is the important point. Cells of heart and brain, they never divide. So they don't undergo any division. Next one is bone marrow. Bone marrow is present in the bones. You might have seen that when you are taking mutton and all, there in the inside that some reddish material will be there. That is nothing but the bone marrow. Actually, this is a seat for the production of RBC. Okay, the lifespan of RBC is 120 days. They show active division, and this is highly regulated. This is a very important point. Cell division should be regulated. Otherwise, say if you take cancer, that is malignance. So here the cancerous cells will not have any control. So continuously, irregularly they develop and they disturb the host other cells, and automatically that leads to death of the individual. That's why the growth of the cell should be regulated, and that is done in case of body cells. That is occurs only when needed. So regulation is nothing but whenever the need is there, then it regulates. Here we can take one example so that you can understand clearly. Bleeding during a cut. If any cut is there, injury is there, then we get the bleeding. But after that, that will be healed. How this is healed? Because of the mitosis. Okay. So, if mitosis is not regulated, the number of cells will be increased. That will be developed continuously, and that leads to abnormal growth of the cells, because when the cells continuously grow, they disturb the growth of the other cells. So, they intervene in the other cells. So, automatically that leads to cancer. Abnormal growth of cells is nothing but the cancer. Okay, that leads to death of the person. That's why that should be regulated. So this is highly regulated in case of somatic cells. That shows the healthy nature of the person. Then cancer cells are not controlled by the growth of regular hormones, regulating hormones. They continuously divide and kill the host. This already I discussed. Now cell cycle. What is cell cycle? Cell cycle, this should be there, and how it will be repeated? Nothing but the cell division is repeated. How this is repeated in detail, let us learn now. The duration of mitosis. Actually, this mitosis is an active division, and here this takes 40 to 60 minutes. Okay, one mitosis period is 40 to 60 minutes. That is nothing but 1 hour. Okay, after this 1 hour, again it takes some rest. That we call as interphase. So during this interphase, that is just the resting stage. So after one mitotic division, it undergoes some rest, and this period is called as interphase, and the period between two cell divisions is interphase. Okay, that is nothing but the resting stage. Next one is during this, actually what happens during this interphase? The genetic material makes its copy to the to distribute equally to the daughter cells. Yes, here DNA is already there. Okay, this DNA will be duplicated in the interphase, because that should be distributed equally to the daughter cells. Duplication is equal distribution. That's why this will be happen only during the interphase. Then after that, this interphase is having actually three subdivisions. First one is G1 phase, S phase, and G2 phase. What is G1 phase? G1 phase is nothing but a linking period between the completion of mitosis. Yeah, two stages are there, no? From one mitosis to other mitosis, there will be some linking. That linking is nothing but the G1 phase. That is nothing but the resting stage. During this, actually, this genetic material transfer and everything will be done. Okay, so this is resting stage between two mitotic divisions. Next one is S phase. What is this? Actually, see here. One more clear explanation here I gave, and the beginning of the DNA replication. DNA duplication will be done. So this is gap 1 phase. Okay, that's why G1 phase we call. Then next one is here the cell also increases in size. Then next one is S phase, synthesis of DNA. So the duplication is nothing but synthesis of preparation of DNA. That should be done. So that is happened in the S phase. And duplication of chromosomes also will be done. These 2 points. These two things will be happened in S phase. Then the following is G2 phase. This is the time between the end of DNA replication to the beginning of the mitosis. So G2. So G2 phase. G2 phase is nothing but the stage between the end of DNA replication to the beginning of the mitosis. That means the stage between mitosis 1 and mitosis 2. That is the G2 phase. Okay. So G1 phase is from first to next, and G2 phase is from second to the following. Okay, between this S phase, that is the synthesis of DNA. Okay, I hope this is clear to you, cell cycle. And if any doubts are there, send me the message so that I will clarify your doubts. Thank you.